Let's discuss on enzymes, their classification. The basis of life is well organized and coordinated network of chemical reactions, and these chemical reactions are catalyzed with the enzyme. So these enzymes are essential for a healthy life. So enzymes being the biocatalysts produced by the living tissues, they are the proteins with the exception of few classes of RNA molecules called the ribozyme. They accelerate the specific chemical reactions without being consumed in the process. They function in aqueous solution under very mild conditions of temperature and pH. The catalytic activity of enzymes depend on their native protein conformation. If an enzyme is denatured or dissociated into its subunit, the catalytic activity is being lost. The primary, secondary, tertiary, and the quaternary structure of protein enzymes are essential to their catalytic activity. So let's move on to the classification. They have been classified according to the type of reactions they catalyze. All the enzymes have the formal EC or enzyme commission number and names, and most have trivial names. According to the International Union of Biochemistry and Molecular Biology, that is IUBMB system, the enzymes are classified into the 7 major classes, and enzymes are classified according to the type of reaction they catalyze. Each enzyme is assigned a 4-digit or EC number, the first 3 digits of which define the reactions catalyzed, and the fourth of which is a unique identifier number. So we will take the example of hexokinase here. It's given the name 2-2-1-1. So each digit indicates like 2. 2 means the second class, it belongs to the class second, that is the transferases. We will see ahead. Then 7 means the seventh subclass, that is the phosphate transferase. Then 1 stands for the first subclass in which the acceptor of phosphate group is an alcoholic group. And again, the 1 is the unique identifier number. It is the first enzyme in the 7th subclass. So 7 classes as per the IUBMB system are as follows: the EC1 oxoreductase, EC2 transferase, EC3 hydrolase, EC4 lyase, EC5 isomerase, EC6 ligase, EC7 translocases. Now EC1 or the oxidoreductases, they catalyze the oxidation reduction reactions. Enzyme in this category include the dehydrogenases, reductases, oxidases, and peroxidases. EC2 or the transferases class, hexokinase, we have seen in the previous slides, is an example of this class. So it catalyzes the transfer of a group such as amino group, carbonyl group, methyl group, phosphate group, and on the basis of which kind of group they are transferring, they are having different subclass. So how they are doing in this reaction, this is the group which is to be transferred, that is X. So from A to B, there is a transfer under the influence of transferases. The enzyme in this category include the aminotransferases or transaminases. Then kinases, they catalyze the transfer of phosphate, that is the transcarboxylase. Then EC3 hydrolase classes, they catalyze the cleavage of CO, CN or CC bonds and some other bonds with the addition of water, because hydrolases are doing the hydrolysis in the presence of water. So this bond is going to be broken up in the presence of water, and now the product are being formed like this, AOH and BH. Enzyme in this category are all digestive enzymes like alpha amylase, pepsins, trypsins, chymotraxins, acid phosphates, etc. Now EC4 or the lyases, they catalyze the cleavage of CO, CC and CN bonds by means of hydrolysis or the oxidation, giving rise to the compound with double bond, or catalyze the reverse reaction by addition of a group to a double bond. So in this reaction we will see This is the A, B, and the two groups are attached here, X and Y. So these bonds are to be broken up, and these bonds are now in the presence of liases, a double bond appear because the valencies are lost. So now it's doubly bonded, and this XY is now separated. The reaction can happen in the reverse direction with the help of the antagonistic enzyme, that is the synthase, and which lead the reaction in the reverse order and regenerating the product. So reverse reaction is important, then synthase or is used in this name. EC5 or isomerase, they catalyze the intermolecular structural rearrangement in the molecule. They are called the epimerases, isomerases, mutases, depending on the type of isomerism involved. So we can see here ABC converted to by shuffling of like rearrangement of the intermolecular structure because of shuffling of bonds. So the arrangement is like CAB, right? Now, next is the ligases or synthetases. The catalyze the joining of two molecules and coupled with hydrolysis of ATP. So it is a synthetases means investment of ATP is required here, energy requiring process, and AB is now being formed. It's joined and ADP is being formed. One high energy bond is being broken here. Then EC7, that is the translocase. This is a newer class, newly added class of the, as per the EC uh, commission, enzyme commission. The translocases catalyze the movement of ions or molecule across the membrane or their separation within the membranes. So examples of translocases are either enzymes catalyzing the translocation of hydrons, inorganic cations, inorganic anions, amino acids and peptides, and the carbohydrates and their derivatives, or the enzymes of the reaction that provided the driving force for the translocation linked to, like oxidoreductase reactions, hydrolysis of nucleoside triphosphate, hydrolysis of diphosphates, and decarboxylation reactions. Thank you.